Good morning, good afternoon, and good evening, everyone. And welcome to this Inside Scientific webinar titled Exploring Estrogen's Role in Metabolism and the Use of 13C-Labeled Nutrients for Advanced Animal Physiology and Nutrition Research. This is Liam Sanyo from the events team here at scientist.com, and I'll be the host today. This webinar has been sponsored by Sable Systems International, so a big thanks to them for helping to make this webinar possible. We're very fortunate to be joined by Dr. Reilly Enos, who is assistant professor in the Department of Pathology, Microbiology, and Immunology at the University of South Carolina School of Medicine, and Dr. Eran Levin, head of the Nutritional Ecology Lab at the Tel Aviv University School of Zoology. All right, and without any further ado, I'm very pleased to welcome the first speaker, Dr. Reilly Enos. Reilly, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you, Liam. Okay, so today, with respect to the webinar, I'm going to give you a brief outline of what I'm going to talk about. I'm going to talk about sex steroids in general, including androgens and estrogens, clinical morbidities linked to estrogen deficiency, and then I'm also going to give a brief overview of some of the questions that elude scientists with respect to estrogen and how we as a group, a lab group, are trying to shed light on these unanswered questions using basic science and animal models. And then finally, I'm going to talk about the translational relevance of our research, just because we want to understand from a basic perspective how we can help patients. So jumping straight into it, we're well aware that there are a number of steroids. We're going to focus on the sex steroids, the androgens, and the estrogens. In particular, we're going to focus on these estrogens and estradiol to be specific. I want to bring your attention to aromatase. CYP19A1 is also known as aromatase. It is important because it is necessary to convert androgens into estrogens. Now, when we talk about sites of steroid synthesis, the predominant sites are the gonads. So when we think of a female, we'll think of ovaries. When we think of a male, we'll think of testes, right? And then the adrenal glands will also synthesize steroids, in particular the adrenal cortex. Now, there are also secondary sites where androgens, for example, can be metabolized into estrogens. Adipose tissue, brain, skeletal muscle, kidney. These are examples of tissues which do metabolize androgens. And to give you a brief snippet of that, for example, you can have androstenedione or testosterone that goes into the tissue of interest and gets metabolized through that aromatase enzyme into estrogen, for example. Now, when we talk about androgens and estrogens, there are primary androgens here, and there are also estrogens, E1, E2, E3, and E4. The primary estrogen that we'll be discussing is 17-beta estradiol, also known as E2, and this is important because it is the primary estrogen during pre-menopause, okay? In humans, for example, estrone is the primary estrogen during post-menopause, and that's primarily synthesized, for example, by the adipose tissue. So when we talk about estrogen throughout the lifespan and we look at the y-axis over here, this is the percentage of maximum through an individual's life. So let's look at estradiol, for example, and this is in red, okay, the straight line here. And as you see, as women transition into the menopausal transition, you see this steep drop off. Now, the question then becomes is, what is this drop-off associated with? What is the problem with this drop-off? And when we look at clinical morbidities, it's already established that there's a higher risk for diabetes in females during that age. And also, there was a clinical paper that was published that followed females through the postmenopausal or the menopausal transition, and they found that yes, estradiol drops, follicle stimulating hormone increased, but they also found that visceral adipose tissue increases and activity decreases. So given this baseline, okay, of information that I've given you, what are some of the questions that elude scientists? Well, how is E2 protective against obesity and associated metabolic dysfunction? Are these effects central or peripheral? Meaning, is the effects of estradiol in the brain driving the outcome, or is it the effect of E2 in the periphery, for example? And then one thing that we're very much interested in is what levels of E2 are necessary to elicit certain physiological effects. Now, why is this important? Well, we know that E2 deficiency, for example, is also associated with osteoporosis. So, for example, do we need a minimal amount of E2 to rescue bone mineral density loss? Is there a certain threshold that we need to reach to impact E2's effects in the brain, or affect inflammation, or affect glucose metabolism? These are questions that we'd like to answer. And in order to do that, we must understand the natural cyclicity of E2, which I'll discuss later in a few slides. So before I talk about these or give answers to these questions or thoughts to these questions, we need to understand what animal models can we use to answer these questions. So the two models that we primarily use in our experiments are the ovariectomy or aromatase knockout mouse model. Now, the ovariectomy mouse model is very straightforward. You simply do a bilateral removal of the ovaries, and this is beneficial, or not beneficial, it's very quick and easy to do, and it produces non-detectable or very low levels of plasma estradiol. The drawbacks of this particular model is the fact that there's no perimenopause, right? You just basically lose estrogen almost immediately. And depending on the age at which you OVX, the duration of sexual maturity can be a factor. So we typically wait until the mice are sexually immature, 10, 12 weeks of age, and then we induce that surgery. The other model is a genetic model. It is the aromatase knockout model. And if you remember in our previous slides, we know that aromatase is necessary to convert androgens to estrogens. So if we have an aromatase null mouse, we should theoretically have no estrogen. So this brings us to the first question is, how is E2 protective against obesity and associated metabolic dysfunction? Are the effects central or peripheral? Now, we know centrally from previous research from established investigators that removal of E2 from the brain, leads to increased food intake and decreases physical activity. So we know that estradiol impacts food intake and physical activity. And peripherally, primarily through deletion or blockade of the primary estrogen receptor, ER alpha, we've been able, or scientists have been able to assess the impact that estrogen signaling has on peripheral tissues. Now, we will talk about data that is not published, but here's an example. So here are two models of estrogen deficiency, and what we have here is female wild type mice, C57BL6, all 10 to 11 weeks of age, and here they are on a low-fat diet. We put these mice on a high-fat diet, and sure enough, they gain weight. And if you ovariectomize and put them on the high-fat diet, they also gain a significant amount of weight, more so than the female wild type alone. Now, what is interesting, if you look at the female arome knockout mice, so these are both low-fat diet and high-fat diet, they start baseline at a much heavier weight than these other mice, okay? However, when we normalize to delta or body weight gain, we can see that the OVX and the aromatase knockout mice gain a similar amount of weight over time. So what is driving this difference in baseline? Well, these mice, they don't move as much and they eat a little more. And because of that, they already at 10 weeks of age show an increase in body weight gain. So we have access to the Promethean metabolic cages from Sable Systems, and this is established, but we show that sure enough, if you look at just meters moved or physical activities, general physical activity in the cage, both the ovariectomized mice and the female arome knockout mice, they move less. Now, if you give them access to running wheels, and if you don't know this, mice like to run, especially during the dark cycle, and we show that wheel running is dramatically decreased both in the OVX and the aromatase knockout mice, suggesting that there is a central effect at play. Also, food intake is also increased in the knockout mice, okay? So given these central effects, How do you tease out central versus peripheral effects? Because the argument can be made that sure enough, if you have these central effects, then and which leads to increased food intake, decreased physical activity, and weight gain, it may be that the central effects truly are the predominant player in regulating the metabolic dysfunction. And it's not necessarily the peripheral effects or the peripheral action of E2 that really is important for the beneficial effects of E2 therapy. So what we did is we designed a mouse model that allows us to inducibly turn on skeletal muscle or inducibly turn on aromatase in skeletal muscle, okay? And when this is the doxycycline induced model, so we treat the mice with doxycycline. When doxycycline is given, it leads to skeletal muscle aromatase overexpression. And we did two different studies, a prevention study and an intervention study. And we did both gonadally intact and ovariectomized female mice. And the purpose of the prevention study was to say, hey, let's give a high-fat diet and doxycycline, turn on skeletal muscle aromatase at the initiation of the experiment. For the intervention study, we wanted to get the mice fat and obese and then turn on skeletal muscle aromatase to see if that would be beneficial as a therapeutic option. The idea being that if you turn on or increase skeletal muscle aromatase, you should then have increased E2 or estradiol in skeletal muscle. So what we found is that both in the intact and ovariectomized models, we did not find any benefit with respect to differences in body weight or body fat percentage between the genotypes. There was obviously a diet effect, but there was no genotype effect, okay? Neither for visceral fat in both the OVX and intact models. Interestingly, however, we did see an increase in uterus in both the OVX and intact models and bone mineral density. Now, I know the question is going to be at what are the E2 levels, and we'll get to that in a second. Now, with respect to glucose metabolism, we did not see any benefit, but with respect to adipose tissue inflammation, we looked at markers, macrophage markers in particular, CD68, which is a pan-macrophage marker, and CD11C, which is a pro-inflammatory macrophage marker in adipose tissue. And we saw reductions. Now, if you're not familiar with inflammation in adipose tissue, so inflammation in adipose tissue is the hallmark of obesity, and the primary immune cells deriving that inflammation are macrophages. So this suggests that in our mouse models, we did reduce adipose tissue inflammation. Now, for the intervention study, again, remember that we got the mice fat first and then initiated overexpression of aromatase. We did not, similar to the prevention study, see any benefit with respect to body composition changes. We didn't even see bone mineral density changes. We did not see adipose tissue inflammation changes, nor did we see any metabolic changes. We did, however, see a change in uterus size, okay? So this leads us to what about the estrogen levels? And the number one issue that plagues sex steroid researchers is the ability to accurately, at very low levels, measure androgens and estrogens in particular. So we have developed a methodology, or we didn't develop it, but we're using a previously published methodology where we can derivatize E2 and measure it at this limit of quantification, okay? So with that being said, we assessed the androgen and estrogen levels, well, testosterone and E2, but I really want you to focus on the E2 levels in these two models. So in the prevention model, we can clearly see that E2 was increased in both the intact and ovariectomized mice, okay? Also, when you just look at wild type mice, you can see the difference between intact mice and ovariectomized mice. So there's a big decrease in E2 in skeletal muscle upon ovariectomy. Surprisingly, we found that this E2 leaked into plasma, leaked into circulation, as you can see here. Now, for the intervention study, we did increase E2, okay, in skeletal muscle, and it did leak into circulation, but it was less than what we found in the prevention study. Now, we didn't publish this in our publications or in our publication, but what we did do is after we gained the ability to assess adipose tissue steroid levels, we then did a follow-up analysis. And what we found is if you look at female high-fat diet wild type in adipose tissue, this is the level of E2 that you would find. Okay. Here's the OVX. It's about 2.5 picograms or lower per gram of tissue. And here's our prevention level. Okay. And here is our intervention level in the adipose tissue. So this suggests that this level of E2 was sufficient to reduce inflammation, but this level was not. Okay. Also, based upon the fact that in the prevention experiment, we were able to increase bone mineral density, right? This level of plasma E2 is probably sufficient to impact bone mineral density, but this was not. Now, granted, it also may be the time or the timing of how long the bone was exposed to this level of E2, but it just goes to show that level of E2 is important to probably impact physiological outcomes. So to go further into this point, we did a follow-up study in males. And so again, we did an intervention experiment, gave doxycycline to overexpress aromatase in skeletal muscle. And surprisingly, or maybe not surprisingly, what we found is a reduction in body weight, improved body composition change, decreased fat mass, decreased visceral fat, liver weight, hepatic lipid accumulation. And if you look at this, it's actually a handout that you have access to, but we show reduced adipose tissue inflammation and improved glucose metabolism. Okay. So why did the males respond better than the females? We have to remember that for this particular model, testosterone is converted to E2. So these are males and they have higher testosterone levels. And because they have higher testosterone levels, you would think they have higher E2 levels. And sure enough, what you found is in the skeletal muscle, we had significantly higher E2 levels in both the skeletal muscle, plasma, and adipose tissue. And if we go back to our previous slide, you can appreciate the fact that in the adipose tissue, you're only getting about 15 picograms here per gram. And in plasma, you're only getting about 26 picograms in the prevention study. And here we're getting a drastically higher amount. And it's likely that this higher amount leads to increase in the brain, which impacts this. And to follow up on that point, we obviously put these mice, or not obviously, but put these mice in the metabolic cages. And what we found is there was a difference in respiratory exchange ratio and food intake, which led to a decreased energy balance. Now, of interest was the fact that the RER was lower within about 72 hours. So we saw changes in RER very early on where food intake was not impacted until after 72 hours, suggesting that whatever happened, the leakage of E2 into circulation promoted fat oxidation or promoted a lower RER, which suggests that there's increased fat oxidation. Okay, we would obviously have to perform follow-up studies to conclude that. But interestingly, we saw the decrease in total food consumed, which led to a negative energy balance. This did not impact physical activity, but it impacted food intake, suggesting that there was a central effect of E2. Okay, so different levels of E2 seem to impact different physiological processes, whether that be in the periphery, bone mineral density, for example, adipose tissue inflammation, or centrally when it comes to food intake. So why is this important for hormone replacement therapy? Well, I will discuss that in the next couple of slides. So what levels of E2 are necessary to elicit certain physiological effects? And I pose this question, but in order to do that, we must understand the natural cyclicity of E2. And if we want to understand the natural cyclicity of E2, we have to look at our models. So when we look at humans and when we look at mice, we clearly know in humans that females go through a menstrual cycle and where we see variations in the level of E2, for example, okay? Mice are different than humans in that they cycle their cycle is about 5 days through different stages. And the general consensus is during proestrus is when you see the highest levels of estrogen, okay? So this, I'd like to say ahead of time, this is debatable because there is uncertainty in the field. For example, David Handelsman, who is a leader in this field, published a paper where he looked at E2 levels and he found during proestrus, this was found to be the highest levels of E2, okay? However, a follow-up study in 2023, Handelsman was on the paper, but interestingly, they found that E2 was highest during diestrus. I encourage you to look at both these papers. I can't explain the discrepancies, but this is the knowledge we had going into our study. So what we did, this is an unpublished study, but we monitored mice, okay? We assessed the stage of the estrous cycle, and we focused on proestrus, estrus, and diestrus 1. On a daily basis, we did vaginal lavage and looked at the cytology of the cells in order to determine the stage of the estrous cycle. And then after 3 weeks of confirming that the mice were cycling, we killed them or sacrificed them between 9 to 10:30 a.m. Mice were sacrificed, we collected their blood, and then we perfused the whole body with max spec grade water, 50 mLs. And then we collected these 16 tissues and examined sex steroids. Now, the uterus weight of these mice followed as expected. Proestrus had the highest wet weight, followed by estrus, diestrus, and then OVX. And then the serum E2 followed a similar pattern. Now, I will show you this data to share with you. It's rather interesting looking at the different tissues. But here you will see that proestrus, estrus, diestrus, and OVX, you can see the levels of E2 for all these different tissues. And what's striking is the fact that proestrus over the course of all the tissues has the highest levels of E2 in every single one, okay? OVX is almost non-detectable in every single tissue. And what was also interesting was the fact that we saw differences with respect to pancreas, for example, has very high levels of E2 during the proestrus, where this falls off during estrus and diestrus. Also, the adipose tissue is a rich source of E2 in terms of content, okay? Now, we've also analyzed other steroids, but I'm not going to share this data. We've also started to analyze the brain, which is also very exciting. Again, I'm not going to show you this data because we're focusing on E2, for example. So what's the clinical relevance of this? Well, think of hormone replacement therapy. There are different modes of hormone replacement: nasal, pills, skin gel, skin patches, vaginal creams, vaginal rings. There's combinational therapies. So all of these therapies and different modes of deliveries will affect bioavailability of E2. So I know that there's weariness to the potential side effects due to whole body effects, right? So potentially the ideal goal would be to get tissue-specific delivery of E2. But the question then becomes is, if we want to do tissue-specific delivery, what level of E2 do we need to reach in order to have physiological benefits, right? And that's basically what my lab or what our group is trying to figure out, because hormone replacement therapy in its current state is a whole body treatment in most cases, okay? And we don't truly understand or don't understand well enough the bioavailability, how the mode of delivery affects bioavailability, okay? So in conclusion, we believe that brain is king. Just by the aromatase knockout, we clearly show, or it's established, that if you don't have E2 in the whole body, you're essentially going to be impacted in the sense that physical activity dramatically decreases and increased food intake happens, okay? And this seems to occur, although the periphery is involved, it seems that brain is the key target. And then inducible overexpression of aromatase in other tissues. This is what we're trying to do. We're trying to look at specific tissues to figure out the role of estrogen in those tissues and the level of E2 we need to provide a therapeutic benefit. So targeted estrogen therapies in the future, what are the needs of the patient? We need to think about mode of delivery, type of estrogen, dose of delivery, when to administer, and combinational therapies. These are all important for developing therapies that are effective and do not provide any off-target effects. So thank you very much. I'd like to acknowledge my current and former lab team, the funding, faculty, mentors, and collaborators, especially Priscilla Firth and John McCarthy, providing some of the unique animal models that I described today. I'd also like to thank my mentors, Angela Murphy, and then Deborah Clegg, who's been a mentor from afar. And then thank you very much. And more than anything, I'd like to say that we are happy to collaborate. We are one of the few labs in the United States to be able to measure E2 in multiple tissues. So if you'd like to measure E2 or other steroids, please contact us. And then lastly, I'd like to thank Stable Systems. Although they have a great product, more than anything, their service is second to none, and we thank them very much. So thank you. And please feel free to ask me any questions at the end of the talk.
All right, Reilly. Well, thank you for a really fantastic presentation. And with that, I will invite Dr. Eran Levin. Welcome. Thanks so much for being here, and you can take it away whenever you're ready.
Thank you, Liam. So I'm a zoologist. It's a kind of extinct animal. And I believe that the truth is out there. I believe that science today is focused on very specific 4 or 5 animal models. And the nature is full of other animals with extreme adaptation developed during millions of years of evolution. And I usually pick my models as their physiology or nutrition is special. We work with shrews, the smallest mammals, with bats, with hawk moths, bees, snakes, lizards, hornets, and I will show you some of our work with this animal, and I will show you work that we do when we use stable carbon isotopes to study nutrition physiology. So first I have to say that during the last decades, the stable carbon isotope analyzer became very compact, very affordable, and easy to work with. And you can use them as classic respirometry to measure a ratio of carbon in exhaled breath and the ratio of isotope in exhaled breath, and also to measure in specific tissues the ratio between the stable isotopes. And I'll show you some implication of it. And in addition, there is tons of carbon-enriched nutrients that are commercially available. Almost any nutrient you can think about, you can buy it when it's labeled, specifically on one carbon or all the carbon, and I will show you some examples. To the ones that don't know much about respirometry, so classic respirometry, you put the animal inside a closed chamber, air comes in, air comes out, and when it comes out, you can measure how much carbon dioxide was exhaled by the animal and how much oxygen consumed by the animal, and you can calculate what are the metabolic fuel that the animal is used, which is the RER, and you can calculate, of course, the metabolic rate. And in addition, today you can add the stable carbon analyzer in parallel, and you can measure specific nutrients that you fed to the animal. You can measure how they go out in the breath of the animal. A more simple setting of this system is to get rid of all the analyzer for oxygen and carbon dioxide and work only with the stable carbon analyzer as also a carbon dioxide analyzer, and assume that you know what is the metabolic fuel that the animal is burning. And I will give more details about it in my talk. To work with tissue, you have to transform them into a gas. So you have to burn them in combustion chamber in oxygen-rich environment. And then you analyze the gas from the tissue by the analyzer. Today, you need a very small amount of tissue for this kind of analysis. 1 milligram of dry matter, it's like the brain of a bumblebee. So we can work with very small animal in very small tissues and still have a signal from them, from your work. And now I'll give you examples how I use in my lab macronutrient. I'll talk about carbohydrates, fatty acids, and amino acids, and I'll give you some examples. Carbohydrates, there are many different carbohydrates that you can buy labeled. You can buy them labeled on specific carbon or all labeled like glucose with the 6 carbon labeled. You can feed them to the animal and then track them when they come out in their breathing, or track them in specific metabolic pathways, and I'll show you an example of something that we did. Here you see the RER for animals that feed on nectar. Nectar is very rich in sugar. These animals usually have a very high metabolic rate. On the left, you see hummingbird, sunbird. It's the African version of the hummingbird, and nectar feeding bat on the right. And you can see the RER for these animals, and you can see that after they feed, their RER is above 1. To the one that are not familiar with RER is usually between 1 when animals burn only carbohydrates to 0.7 when they burn lipids, and in the middle is a mixture. And for nectar feeding animal, you see that RER goes above 1. It's known for more than 100 of years. In the literature, it was written that it's because of they synthesize lipids, but when you go to the metabolic pathway of lipid synthesis, there is no extra carbon dioxide when you synthesize lipid. So we worked with hawk moths. Hawk moths, I like them because they have the highest metabolic rate known in nature. And when you feed a hawk moth and you test for his RER, you get a very high value, above 1. And in the beginning, we thought that we have a problem when we measure this very high RER. We test it also for monarch butterfly, again, very high RER. And to be sure that we are doing the measurement right, we test it for a bee. Bees always burn carbohydrates, and they should have RER 1. So when we measured it for a bee, we found that our system worked properly and calibrated, and we get very high RER for the other animals. So what can bring us extra carbon dioxide? We start to go into the biochemistry, and we found that when you activate the pentose phosphate pathway, in the oxidative phase of the pentose phosphate pathway, when glucose turns into ribulose, you get the carbon number 1 from the glucose goes out as carbon dioxide without consuming any oxygen. So it's a potential pathway that can explain the very high RER that we get for nectar feeding animals. So what we did next is feed the hawk moths with 2 different labeled sugar. First, we fed them with a glucose labeled on carbon number 1. So we expect that if the RER goes down, there will be less labeled carbon, the delta C13 will be lower. And if we feed then the hawk moths with glucose labeled on carbon number 2, when the RER go down, we'll have more labeled sugar because we'll have less unlabeled carbon from carbon 1. And I'll show you the results, and it will help you to see. Here you see in black the RER, and you see that when it goes down, also the delta C13 goes down like we expected when we labeled and fed them glucose labeled on carbon 1. When we fed them with glucose labeled carbon 2, we got exactly what we expected. When the RER goes down, there is more labeled carbon from carbon number 2. And we can say that the high RER is because of the use of pentose phosphate pathway that we see in these animals. And in our paper, we explain why it's important for nectar feeding animals, but this is for another talk. We can also test complex sugars, the most common polymers in nature, cellulose and chitin. Cellulose, you can buy them labeled online. Chitin, we cannot find them anywhere to buy labeled chitin. And we wanted to know if hornets can break chitin. Hornets are very interesting animals because they hunt for other animals or eat carrion. They bring it to the nest and they feed it to the larvae. And the larvae break the protein and the skeleton of the insect and feed it back to the adult. To test if hornets can digest chitin, we rear beetles on labeled glucose for 1 month. Then we extracted the chitin from their exoskeleton, and we fed it to hornets without larvae and to larvae. And then we tested their breasts in the carbon analyzer. You can see that for the adult hornet, they couldn't break any of the chitin, but you can see that for the larvae could break the chitin and they can digest it. Later, we tested the microbiome of the larvae, and we found 3 bacteria that are suspected to be able to break the chitin for these insects, and they're absent from the adult, and it might explain how they can break the chitin, which is a polymer that most animals cannot break. We also tested ethanol. Ethanol is very popular now in study in insects and in mammals. 5% of all deaths around the world are because of abuse of ethanol use. And we test if hornets can break ethanol, because it's very known that hornets attract to ethanol, and there are a lot of funny news about ethanol crazy hornets in Europe especially. And in this experiment, we fed ethanol labeled on carbon number 1 to a hornet and to a bee and put them in a metabolic chamber. And you can see the rate of oxidation of the ethanol by the hornet, which is extremely fast, while the bee very slowly metabolize the ethanol. And it might explain why bees get poisoned by ethanol and also get addicted, while hornets are not. And it's a project that we are working on right now. I really like fatty acids. I think they have a lot of implication on ecology and in physiology of animals. And as you know, there are saturated fatty acids that usually, if you find it in plant in more warm area, and unsaturated fatty acids that more common in cold areas. And a lot of people think about good fat and bad fat. I don't believe in this kind of thing, but there is a very big importance to understand what is the ability to assimilate this fat. And to test the assimilation of this fat, we tested 3 fatty acids: palmitic acid, linoleic acid, and oleic acid. These are the 3 most common fatty acids in our diet and in animal diet. A palmitic acid is saturated, oleic acid is monounsaturated, and linoleic acid is polyunsaturated with 2 double bonds. We tested on 2 vertebrate models, 2 insect-eating vertebrates. First, it's the pygmy shrew. It's the smallest mammal in the world with the highest metabolic rate, feed on insects. The second is a gecko, the same size as the shrew, but a reptile, and also insectivore. And the third one is a bumblebee, a nectarivore that feeds also on pollen. We fed this animal with one of the 3 labeled fatty acids. And for the gecko and the shrew, you can see that the maximum delta C13 was more or less the same for the unsaturated fatty acids, but there was no oxidations of the saturated fatty acid. And when we test their feces on the right, you can see that most of the palmitic acid was excreted in their feces. So there was no absorbance of the saturated fatty acid. We found more or less the same for the bee. You see on the right, in the respiration, bees oxidize the oleic acid and linoleic acid in high rate, but not palmitic acid. And when we tested their tissues later, we found the linoleic acid in the fat body and the oleic body in lower rate in the fat body and other part of the body. Very interesting finding that we found. It's now in preparation. We found that the linoleic acid that considered essential when the bees feed on this fatty acid, they saturated it back and accumulate in the body as saturated fatty acid. Amino acids and proteins are also available, especially amino acids, not just protein organic amino acids, but also special amino acids like GABA, or beta-alanine, and taurine. And we did some experiment using these amino acids. And I'll show you something interesting that we did with hornets again. As I told you, the common knowledge is that hornets cannot break protein. They have to bring it to the nest, feed it to their sister that are larvae, they are sessile. The larvae, by gluconeogenesis, make the protein into sugar, and they give back to the adult hornet a drop of liquid that is very rich in sugar and in amino acid, which is especially proline, leucine, and glycine. A few years ago, somebody took this composition, make an energy drink, and everybody is using it today. It's very efficient, but nobody ever tested what is the efficiency, why it's so efficient. So we try to see what happened with these amino acids in the hornets. We make artificial larvae secretion that each time we labeled one of the amino acids, proline, leucine, and glycine, we fed it to the hornets, and then we tested in their breast what is coming out, and in their brain, muscle, and fat body, what is the accumulation of the amino acid. Here you see on the left during rest, the oxidation of the amino acids, on the right during flight. So you can see that the 3 amino acids are oxidized during rest, but during flight, only proline oxidized in very high rate in this hornet. And when we test in their tissue later, we see that there is no incorporation of proline at all into this tissue, only incorporation of the leucine and the glycine into the tissues in every tissues that we test. And we can say that the proline is oxidized as metabolic fuel, and probably this is what gives the extra energy through proline dehydrogenase in the body of these insects and give them more efficient performance of the mitochondria. We also challenge the common knowledge that I told you about the possible of hornets to break protein by we reared labeled protein. We fed bumblebees with leucine labeled on carbon number 1. Any modification of the leucine will throw away the labeled carbon. So we can be sure that every labeled carbon that we find in the body of the bee later is related to a labeled protein. Then we fed these bees into hornets with or without their larvae, and we test the muscle of these hornets after 10 days. And what we found that hornets can break protein. Here you see for queens, workers, but males cannot. And when we test for the hornets with the larvae, we can see that males depend on the larvae to break the protein, and workers break more efficiently the protein when they have the worker, but they can break also the protein by themselves. And the last part I'll show, it's about a free flight metabolic rate. You know that you can measure metabolic rate in a metabolic chamber, and you can measure metabolic rate by doubly more energetic expenditure, but doubly labeled water, but this is for a few days usually, or more than a few days. But if you want to measure the metabolic rate during free flight of an animal or during a sprint, it's very difficult because you can't make them run inside a metabolic chamber or only on a treadmill, and it's not mimic the real life. So we modified a method to inject labeled bicarbonate into the insect. Here on the left, you can see the curve that you get of the delta C13 after injecting the sodium bicarbonate. So after a few minutes, it's getting even in all the body, and you get the peak, and then you get the depletion of the bicarbonate as carbon dioxide from the body of the insect. You can calculate the rate of the depletion of the bicarbonate and plot it against the known metabolic rates of the insect and calculate the rate of metabolic rate. So what we did, we injected these beetles with labeled bicarbonate. We put them in metabolic chamber. We got the slope of the elimination of the metabolic rate. Then we took them out of the metabolic rate, put them in a wind tunnel, let them fly for a few minutes, put them back in the metabolic chamber, and measure again the rate of depletion of carbon dioxide. You can see here the missing parts. You can calculate the slope between before the animal starts flying and when it's end flying. And from the slope, you can calculate the elimination rate of carbon dioxide and plot it against the curve and get the metabolic rate during flight of the animal. You can do it with birds, with bats, and with any other animal too. So to sum up all these examples that I show you, there is endless application for the use of delta C13 labeled macronutrients. We really just start to discover all the potential of these methods. You can study all organism physiology or specific tissue. You can take it out to the field or do it in the lab, and it can answer a question in physiology, metabolism, behavior, nutrient flow, and it's very easy and simple to operate. And with that, I want to thank first my great lab members that did most of the work that I showed you, a stable system for the equipment that we use, and for you for listening.
Fantastic. Well, thank you so much, Eran, for the great presentation. We'll move on right to the Q&A. So, Reilly, I'll invite you back onto the audio and video line. We can get started here. So, Reilly, maybe we'll start with a question for you. Can you maybe expand upon the differences between human and mouse estrogen deficiency?
Yeah, can you hear me?
I can, yeah, loud and clear.
Yeah, so one of the things, and I've been looking at the questions that have been coming in, a major difference between humans and mice is the fact that mice, in terms of peripheral aromatization, they do not aromatize. They don't have a large expression of aromatase as do humans. So one of the questions was with respect to brain aromatization. So we don't, the mouse model we presented was a whole body aromatase knockout. So it's very difficult to compare mice and humans with respect to peripheral aromatization because they don't have that much expression.
Excellent. Great answer. Eran, next question here. So obviously insects have pretty different physiology from humans. Can we really learn something from studying their physiology, from studying these insect models?
Actually, I think that we have a lot in common. We have the same metabolic pathways. And for example, for understanding the pentose phosphate pathway in nectar feeding animals, we can learn about and understand people that have deficiency in this pathway in their body, and there are millions of people around the world. It can help us to understand why they suffer from this because of oxidative stress that and nectar feeding animals develop this to protect from oxidative stress. And there is much in common, but I think that science today is not very integrative, and we work in different fields, and usually we don't do this integration, and I think this is a big problem in science today.
Yeah, definitely. I mean, I have seen a bit of trends lately where there are more and more systems biology approaches where there is a bit more integration, but I think there's still a long way to go, and I think there's a lot more that we can learn from these kinds of approaches. All right, Reilly, maybe one for you. What other thoughts or insights can you give us regarding hormone replacement therapy?
Yeah, that's a great question. So some of my slides were cut off at the end, but I talk about the fact that there's many different modes and methods of delivery for estradiol. Skin patches, orally, injections, gels, creams. All of these things are going to have different bioavailability. In our research, we find that if you give E2 chronically, we don't see an effect with respect to physical activity that we do, but we do see an effect with food intake. So whether you're giving a chronic E2 dose or a pulsatile dose, similar to what we see in physiology, I think these are going to have ramifications for hormone replacement therapy.
Fantastic. All righty. Eran, and we could probably actually question to Reilly as well, but how complicated is it to operate these analyzers?
Actually, I really like this analyzer because I need like 2 hours to get an undergrad student, be a professional working with this analyzer. So they are very simple, very hard to make damage to the analyzer. And it's a very, you can take it out to the field. They're sturdy.
Excellent. And Reilly, what's your experience with them?
Oh, they're very straightforward, very easy to use. More than anything, the service. I mean, if you guys, if we ever have issues, which we don't, we've had issues with mice chewing things sometimes. That's just because mice are. Stable Systems is on the ball. They're very helpful, and their technical support is second to none.
Awesome. Yeah, so great to hear. An interesting question here is, can changes in the E2 levels in tissue plasma be correlated by body weight or changes in by body weight or change in body weight? And if so, how can that be accounted for to ensure the changes in the E2 aren't due to the weight? So Reilly, that one would be for you.
Yeah, that's a great question. So one of the issues, as I alluded to in my talk, is the fact that it's very difficult to measure E2, right? Over the course of a lifespan, for example. And obviously we'd like more human data, but with respect to a mouse, because of the fact that there's very little peripheral aromatization, it's very difficult to examine E2 changes over the course of the lifespan and over the course of weight gain. We've done that a little bit in our work, showing that sure enough there's changes, but also a paper just came out suggesting that what you eat or your diet, the dietary component with respect to progesterone, can actually impact steroid physiology. So there's a lot of factors that go into that.
Excellent, great answer. Now, Eran, one for you. The insect measurements, were they done with a continuous or a stopped flow setup?
Usually I prefer to work with continuous, but sometimes, for example, now I'm working with termites, and they are very, very small, and their metabolic rate is low, and I have to work with stop flow. But you can work both in both ways.
Excellent. Reilly, one for you. So since E2 is lower over time, does the expression of the ER alpha or beta receptors follow? And if not, why?
Another great question. Dr. Heavener from UCLA published a paper looking at adipose tissue expression of ER alpha and she found that it was lower. I believe this is correct. It was lower in females that have poor metabolic outcomes. Okay. The difficulty, again, with this field is the fact that measuring tissue levels of estradiol or estrogen and associating that with a specific expression of the receptor. Because usually we can get the receptor, but due to limitations, I mean, there's only a few laboratories in the world that measure actual tissue levels of estradiol.
Excellent, great answer. All right, one for Dr. Levin. So the extremely high respiratory exchange ratio of the hawk moth is really impressive. Can you discuss the evidence that this is caused through the PPP and other PPP inhibitors that can normalize RER, and are these toxic to these insects?
It's a good question. Actually, we only did like the very strict experiment with the labeled glucose, and I think it's proof behind any question that this is what caused the IRER. Why they have this IRER? It's other part of our work that published, and we suggest that it's to build antioxidant potential in the muscles of the moth, and we showed it in other methods. But we never try to give blockers, and there are tons of things that can be done. But we just show that this is what gives this IRER.
Excellent. Another question for RER, but for you, Dr. Enos. So looking at the variation of RER under E2 treatment, can we conclude that we have a higher metabolic flexibility?
I don't know if we can conclude that. We're still doing ongoing studies right now where we're actually pair feeding mice to determine if there is an improvement in metabolism independent of a body weight change. Now, we didn't show a body weight change in that data set, but the short answer is we cannot confirm that. We're still trying to figure that out.
Excellent. All right, Dr. Levin. So is there any other types of instrumentation that's necessary to measure the levels of accumulated 13C from carbon dioxide in these insects?
For solids, you must use a combustion chamber, which is quite simple and straightforward to operate. Just burn the sample in very high temperature with oxygen, pure oxygen, and then it goes through the analyzer.
Fantastic. I think in the interest of time, we'll just ask one last question, and maybe Reilly, we can start with you. But what kinds of exciting research do you think is coming up? What are the sort of big developments that are coming up maybe in the next 5 years in this field?
I'm hoping that we get more. First, there's the Women's Health Initiative really hurt people or hurt hormone replacement therapy with respect to it gave it a bad rap because I think the data was misunderstood. I think that the moving forward is better assessments of E2 levels and then hopefully in the future targeted E2 delivery therapies. That would be the goal.
Fantastic. And Eran, from your perspective, what do you think are the really exciting developments coming up in the next few years?
I think that people will start to realize how simple and cool this method, and you can apply it on almost every field in zoology, behavior, physiology, and it's really easy to use, and it will give you clear results.
Yeah, very cool. All right, thanks. Well, Eran and Reilly, thanks so much for joining us. It's really been a pleasure having you with us.
Thank you.
Thank you.
Big thanks also, of course, to the audience for joining us today. And last but not least, we'd also like to thank Sable Systems for sponsoring this event. And in closing, we hope you enjoyed this Inside Scientific webinar, and we'll see you again next time. Have a great day, everyone.